Hello everyone, today I will explain to you about genetic variation on drug-related genes. Let's get started. First of all, you need to know what is genetic variation. Genetic variation is variation in DNA sequence between individuals in a population. What are the sources of variation? They are recombination and mutation. Next, what is Exome Aggregation Consortium, or XAC for short? In this study, XAC from 60,706 individuals is used to determine allele frequencies of very rare variant in subpopulation, and comparison is made between them. It is used to determine the extent of genetic variation in human population. Now, what is functional variant? Functional variant is genetic variation which alters gene function. It exists in low frequency in a population, and it may play a role in the development of complex diseases.
Moving on to the definition of germline variation. Germline variation is the alteration of genetic information in reproductive cells, which are sperm and ovum. This mutation can affect directly the treatment outcome of a patient.
There is a study called genome-wide association study, which is used by scientists to study genes involved in human diseases. This method searches genome for single nucleotide polymorphisms, or SNPs for short. SNPs usually are considerably abundant in people with diseases. SNPs already being utilized to study certain chronic diseases such as diabetes and Crohn's disease. However, this technique cannot detect variants with functional effects.
Next, cumulative allele frequencies, or CAP for short, is used to determine number of functional variants and their allele frequencies. Genes that have a high CAP score are said to have high probability of being affected by functional variant. In this study, genes that have a high CAP score are absorption, distribution, metabolism, elimination, and drug target genes. When CAP is combined with drug risk probability, or DRP, it can identify how much a drug can be affected by functional variant. High DRP score indicates that there is an increased chance to carry functional variant, which will alter the outcome of treatment later on. Examples of drugs that have a high DRP score are paclitaxel and docetaxel. However, even though a drug has a high DRP score, certain proportion of patients do not respond properly to the given treatment. This is mainly due to number needed to treat. Number needed to treat is the number of patients needed to overcome one additional bad outcome, such as stroke or death. For example, if the number needed to treat is 8, then 8 patients are needed to prevent another bad additional outcome.
Geographic location also plays a role in contributing to the difference in frequencies of genetic variants. Genes that show the highest difference between populations in this study are cytochrome P450 and phase 2 enzymes.
You have come to the end of the video. Thank you.